Welcome everyone to today's training session on Introduction to Seurat. So before we begin, make sure you download the data sets in the background as well as all of the module scripts. So today we're going to talk about Introduction to Single-Cell RNA-seq Data Analysis using Seurat. I'm Yujie Guo. I'm a research assistant from Heng Li's lab. So here's a basic outline of data of single-cell RNA-seq data processing using Seurat. We start with the sequencing reads, and then you generate the count matrix from the input. For the count matrix, you need to filter the cells using quality matrix, aka QC. And after QC step, you normalize the data and scale it and regress out unwanted variation. In this step, you might also need to integrate some of the process data as well. So you integrate the samples together into one aggregated data sets. Well, then you do unsupervised clustering. And after that, with the marker identification and potentially the cell type assigned, you can then do the trajectory analysis, differential expression of cell types or genes between sample groups, and custom analysis to your fit. So for today's outline, the pre-processing step session has been covered by Dr. Jeremy Simon 2 weeks ago, or for 3 weeks. So today, the first thing we're going to do is learning how to read in different data formats provided by 10x genomics, cell arrangers, and so on and so forth. And then we're going to do a data processing standard workflow. Within this, I'm going to also introduce a method called Doublet Finder that can detect the doublets or artificial doublets in the data sets. After that, we're gonna do a data sets integration. There's two ways of doing, there are multiple ways of doing this. I'm gonna introduce two: harmony and the canonical analysis using Seurat. And then with this in mind, we can then proceed to find markers and cluster identification. We're gonna do this using some of the basic manual ones, as well as I'm gonna introduce one method of automatic cell annotation. And if time allows, I'm going to do a pseudo bulk differential expression analysis using annotated cell types and processed data sets. So, okay, we can begin. If everyone have downloaded their data sets, we can move on to the first one. So 01 data formats.r provided on GitHub. Can people show thumbs or if you can download the data sets before we proceed? Okay, I've seen one thumbs, two. Okay, that's good. Thank you for responding. So here, so after you have the data sets as well, you can go ahead. I think I provided .tar .tar.gz, this one. So you can go ahead to do tar dash, on this data sets, which will provide you with this folder. So, and for me, I'm just gonna directly go to my data sets. You can view, there's several data sets we're gonna play with. So they all comes in different nature. Some of them have different file format. Some of them are integrated data with data from different samples and treatment groups. Okay, we're gonna get started. So before going this, sorry, I forgot. I'm gonna introduce briefly about all of the input file formats that's common for the Seurat analysis. There's the HDF file, which is from the cell arranger. That's the most common one you use for Seurat analysis. There's also RDS file and data object. That's usually used for ScanPy and Python, but you can also convert it for Seurat analysis. There's also Loom. Loom is basically HDF file, but they have additional row and column attributes. And the MTX is a text-based market exchange format. So here, this is a feature barcode sparse matrix. So the reason it's called this, oh, by the way, the column and the rows should exchange with each other usually in Seurat. So feature is just different genes. It might be a feature for a different cell type. The barcode here, it means that in the Seurat object format, the cells are usually identified by their UMI, so like unique barcode. And it's called a sparse matrix because usually, or most cases, all of the genes expression levels are 0. So the matrix is very sparse. Okay, let's get started with the R. So first one, we're gonna read in a RDS format. So we can name it as RDS object. And then, so we're gonna use a method called read RDS. And here we have, so yeah, before moving on, you're welcome to set your working directory to a folder that contains both your R scripts and data sets. Okay, so we go into the data sets and there's one called F and IFNB harmony. S, go ahead, run that. Yeah, if you didn't install the library yet, this is a very quick step. You can just install it using the remotes as well from the GitHub. Right, so we go ahead, run it. It says I don't have, oh, data sets. Yeah. So here you can see that in the RDS format will directly give you a Seurat object. And you can kind of check if the object has been imported. Oh, sorry. View data. In this way, you can sort of see what I was talking about of the feature barcode sparse matrix. These are the individual cells, and these are just the metadata. So this is the immune control, and you have the count, also the stimulation, aka treatment group, separated into control and treatment or stimulated. And this one, you can see it already comes with Seurat annotations. So keep in mind that this is a processed Seurat object, but also at the same time, RDS could just be any unprocessed sparse matrix. Okay, this is the RDS file. Feel free to stop me if you have any questions throughout the process. And next one would be the MTX format. So here we can name it as MTX object. And here I'm going to use a function called read MTX. So before moving on, it has 3 parameters you need to put in: matrix, features, and barcode. Oh, sorry, cell. So MTX matrix usually comes from a tar.gz file. So if you can then move on to the terminal, we're gonna use something called a raw feature barcode matrix tar.gz with the 10K PBMC generated by the next-gen Chromium. You can go ahead to do the So you untar it, it'll give you, so if you to a list, it'll give you a new folder called raw feature barcode matrix. You can just quickly go in to see that it will give you 3 files: matrix, features, and barcodes. So we're gonna put them in here. So we're navigating to data sets, and then what is it called again? Raw feature matrix. The first one would be the matrix. mtx.gz, so go ahead and put that in. Good. Put them all in. It's called barcodes TSV and feature TSV. Feature TSV. And barcodes or barcode, barcodes. Let's see, you can run. Yep, so that's how you would import MTX matrix format. And then, so it's still kind of loading. So when you load an MTX matrix, it won't directly give you a large Seurat object. So you need to sort of create the Seurat object yourself. So since it is a PBMC sample, so I'm gonna call it PBMC Seurat object. So you're gonna do the classical create Seurat object. So it will give you a count matrix. So you can directly put that in, counts equals to MTX object. So this is how you import an MTX object. The next one is a little bit uncommon. It's called Loom file. There's also a tool that's provided by the same lab that created Seurat. They have a Loom R package, but here I'm just going to show how you import a Loom file as well. So here you have something called connect function, and then you provide the file name. So you're going to use a Loom file here, the adult ham organs 10x bone marrow. And then we are going to assign it to the Loom object. Okay, oh, it's already done. That's fast. And then here we are gonna transform it into a Seurat object. So we have a function called s.Seurat, and okay, so right now the matrix file has been done, its uploading and everything. So you can see that this is a large DGC matrix instead of a Seurat object, and then you transform it into a large Seurat object. Okay, I'm not gonna wait on to, I have a MacBook Air, so it's gonna be very slow for me to finish running certain steps. We're gonna move on to the H5AD format. This is the classical one for the ScanPy Python data format. So we're gonna do something called convert. So we want to convert this H5AD format, so we can find it here. It's gonna convert it to a H5 Seurat object, right? So we have the data sets. Then go ahead and copy the H5AD format. And then the destination would be, can call it H5 Seurat. And then you can either set the override parameter into either true or false. It's still loading my matrix. So yeah, this is what you would do to convert and data format into a Seurat format, and then you can just load the H5 Seurat object. So we are gonna use something called load H5 Seurat, and then you can go ahead to copy paste that one, and then it's instead of the and data format, it's gonna be a Seurat format. That was really slow. So Yeah, I'm gonna stop this here. We're not gonna use the Lumar object for the rest of our workshop. Oh God. Okay. So have people have difficulties following me, or is this a good pace so far? Shows of thumbs if you like you're following and everything.
Everybody's trying to find the thumbs.
Everybody did.
So are you?
Oh, we got a thumb.
Oh, okay, thank you.
I'm fine. I'm just having issues here, but I'll follow you. I'm following.
Okay, and load everything. Sorry for the crashing. Hopefully it won't happen soon. Especially with the Zoom on, it's taking resources. Yeah, okay, so the convert works fine. And then we can load the H5 Seurat. Oh, well, you need to assign it for okay, so this is for the and data format. And finally, the most classical one is the 10x cell ranger HDF file format, or we call it H5 format. So this we're gonna use the read 10x H5 10x H5 method. File name is we're gonna use the H5 data sets. For now, we can use the non-small cell lung cancer. Okay, and then we can assign it to non-small cell lung cancer. So this is not yet a Seurat object, but rather a sparse matrix. And then probably your computer runs faster than me. So for the sparse matrix, we can check it using something called str. So it'll give you all of the kind of data framework or format. So here you can see there's gene expression level or RNA data. There's also a column called antibody capture and multiplexing capture for further analysis. For the sake of this, and for the sake of this workshop, we can kind of subset it into only the gene expression. So this will give you the counts. Yeah, so make sure whenever you load H file format, check if it has multiple columns of a different kind of data stored. And so you want to subset into the gene expression for further analysis. So we can check this counts, can quickly check it by subsetting the first 10 by 10. Yeah, here you can see the 10 column names corresponding to the cells, and the 10 rows here are the row names of genes. Okay, and then with this counts, we can further initialize the Seurat object within this raw non-normalized data. So we're gonna do the create Seurat object. Counts equal to counts. So we can also name it with a project that will help you with organizing with all of the data you have. Usually when you have a research project, you will deal with multiple samples. And you can also do an initial processing of have the cells of minimum expression counts of 3, and each cells have at least a feature of 200. So it has 200 genes captured in it. And we're gonna name it to the scalc.Seurat object. And you can quickly check it after it finishes. So yeah, here you can see it's an objective class Seurat. Okay, so this is the first step of data formats input with different formats, and we're gonna moving on to the standard workflow. I don't have any slides for it, so let's go straight right into it. So it's gonna use the latest data. We load the scalc object here, and we're gonna just load another library, Tidyverse. Okay, so we already load the Seurat in the previous step. Now we can quickly view the metadata here. So different cells, it has the original identity from a bulk sample. It's not yet annotated, and we have the count RNA and number of features. So, okay, for this step, we might want to see how compute the mitochondrial contribution per cell and filter out the poor quality cell. So this is already done for you. You can go ahead and do that. And let's view the metadata again. See what happens. So here, it already captured the percent of mitochondrial leads in here. So this is a regex of capturing any gene, any feature name that start with MT. That's going to indicate, oh, this is mitochondria. Okay, and then we can plot an MT violin plot. Okay, so here you can see the distributions of all 3 criteria we're gonna do QC on. So you can see that it's pretty much sparsed out, and it's not telling you much information if you plot it individually. So here, there's a function called feature scatter that can tell you what's the correlation between the two. So to do that, you can do feature scatter. So following this Seurat object, and we can set feature 1 to end count RNA and count underlying RNA and feature 2. You can set it to % mitochondria or others. For now, I'm gonna show you the end feature RNA. For the QC step, it's important you play around with the data to really make sense of the poor quality here. So now I'm gonna add a fitted line just to smooth. So we can do a linear line here. So ideally, you want end count RNA versus end feature RNA fitting toward a straight line. If a cell, so in each dot here represents a cell. If a cell falls here, it means that this one gene of the cell has been sequenced over and over again. So it might be an artifact. So you don't want much dot here. Similarly, you don't want much dot here, which means that this cells, a lot of the genes have been sequenced poorly. So the coverage is very low, the end count RNA, whereas this cell, a lot of genes have been captured, but the sequencing depth has been poorly done. So here, yeah, you can kind of set your subsets criteria or threshold here. And for now, we are just gonna do the filtering step. So yeah, this is what I choose, but feel free to choose a different one depending on the performance of your data sets. Okay, and then after the filtering step, we can start to normalize the data. So here is a function to normalize data. We're gonna do log normalize on all our counts, and the scale factor is 10,000, so it's gonna multiply by 10. So this is a default setting by Seurat. You can feel free to change that, but you can just run the one line normalized data. I'm just gonna check this really quick to see all of the metadata. So here we've performed multiple steps. It will all be captured here. You can see that the count, there are 3 types of parameters. One is counts, and one is data, and one is cells. The counts are the raw one. It'll always be there no matter what steps you process. And the data here is the process data. So you would want to use the data instead of the counts. Okay, after normalization, you can then do the identifying variable features. And then we can sort of see how the top 10 features look like. So we can do head features, it save here in your Seurat object. And then, so with the top 10 features assign it to this variable, we can then plot, let's name it plot 1. Variable feature plot. And then you want it labeled with the top 10 features label points. There's a method called label points. Plot equals to plot 1.
So we're plotting them all, not only the top 10, right?
Yes, yes. So we want to label
Got it.
all of
Yes,
the
yes, yes.
points. Top 10. Okay, so we can see now of this is very detailed analysis because you will need us when because I've checked the Seurat vignette published by their groups is not very well detailed, but instead of following all of the steps using the default Seurat parameters, it might be better if you check the data for yourself. So here you can see that this is the average expression of all of the genes, and this is the standard variance. You can see there are a lot of highly variable genes among all of the cells here.
So variable means variance more bigger than 220.
So we're only taking the top 10, so it's ordered. So regardless
right.
of
Okay.
the actual
Sure, yeah.
variance. Yeah, but you can definitely go check in your detailed Seurat object with all of the variance. Okay, so we have this plot now. Let's go ahead. We can then do the scaling. It's gonna take some time for me for scaling. So any questions so far for the standard workflow on one sample data sets?
I think everything that's on the screen.
It doesn't speak.
Yeah.
Okay. I lost it.
Yes.
Crashed.
Technology, right?
Sure.
Yeah, like my R studio also crashes on me multiple times. Don't crash. Okay, so we're at the scale data step. Just got also gonna. Yes. Yeah, for here, you can see that when I scale the data, I select the features to all of my genes that's included. But when I run the PCAs, we essentially want to do linear dimension on the really variable feature and not including all of the genes that has been consistently expressed throughout different cells. So here I'm subsetting the features to the variable features selected by the find variable feature method in Seurat. While it's running, I'm just gonna go ahead to put in the codes to visualize the PCAs. Usually in a really basic PCA, you would select the first two PC, but that would not be the case for a really complicated single cell analysis. So here I'm just gonna oh, it's still running the scaling. So you can sort of print A. Oh, it's finished running. And then you can print out first check the few dimensions you want to do. Let's like for now do 5 here and features. So it'll print you how different features contribute to the projectings of the PCs, how it's been projected back to the PCs. You can see whether it's positive PC correlation or negative ones. While it's still running, next I'm gonna do something called a dim heat map method. And gonna explain all of this later, while it's finished running, but feel free to put in the code now. Step. With the Zoom, it's more slow for me. So you can see how this function essentially tells you how different genes from the different cells contribute to the first PC here. You can also plate it into how to projecting onto the PC2, PC3, PC4, PC5, and so forth and so on. And this is investigating on individual PCs. And this one will just tell you how the PCs have been projecting with the classic bubble plot. Sorry, it's not responding. If it's like your computer is faster, feel free to kind of explore these functions on your own. This will help you to explore the QC a little bit and determine the best parameters for your Seurat standard workflow. Have people are people following this standard workflow? Am I going too fast, or did you finish running the PCAs? Show of thumbs if you finish running PCAs. See one thumb from Diana. Or which step are you all on right now? Yeah, I want this workshop to be a little bit more coding so that you really understand what's behind the code instead of just copy paste everything and then change your object name. Yeah, so I want everybody, I hope everybody can follow here. Still kind of stuck. I'm gonna move forward to explaining the clustering part. So after you have your PCA results, you can use, oh, I have chat here. Yeah, and it does have a high computing cluster. My main language is Python, so I kind of use it. Are you inside Dana-Farber? If you're inside Dana-Farber, I believe you can use it. If you have like high computing cluster, it'll be definitely faster for you. Okay, oh, great, it's
Got it.
finished running. Okay, yeah, we can then print, okay, it runs. So it shows you the top 5 genes of the top 5 PCs, whether it's being contributed positively or negatively. It's really useful
Nice.
to kind of dissect the whole PCs, because sometimes if you're on Twitter, single cell RNA-seq have been bashed recently. But yeah, so really know what's going on behind all of the statistical number. Which step am I on? Oh, here. And then you can also plot a heat map here. For the PC ones, it'll show you the how much the 500 cells and what each the top expressed genes have been like for the top 500 cells. Okay, and then you can also plot the elbow plot. You can see it's kind of like plateaued here around 15 starting here, so I'm not going to use the rest of the PCs. That's how I'm going to choose dimensionality equals to 15 for clustering. It's going to take some more time. Yeah, after you find the neighbors, there's a really important parameter called resolution when you do the find cluster. So the higher the number, the more cluster assignments you're gonna get, or more potential cell types you're gonna have. So you can, in this step, you can sort of try out different resolution and see what type of resolution works for your data. And so, am I running too much, any resolution? Yes. Hopefully not. And so you can sort of view, it's gonna show up in 5 new columns for your data, and we can sort of see how it's been show up here. And in this, each cell is just mean the cluster assignment of different cells to a potential cell type. You can also plot dimensionality and see how the highest resolution have a lot of cluster assigned together. So we don't want that. And let's see what the 0.1 looks like. So the 0.1 has been a little bit more clearer. So this is a PC plot, not a UMAP or t-SNE, so it's still quite linear-like. You can now look at the idents assigned automatically by Seurat. You can see how it's been so many cluster. So we also don't want that, so we can assign it to 0.1. And then you can check the idents again here. Oh, sorry. Oh God, it should be RNA-seq res 0.1. Yeah. RNA. and after that, just gonna see again. So sorry. Here, you can have the 7 clusters you want to do, and the rest is just running your map. Okay, so now we have the standard workflow. You can see after you map, it's just gonna show you in a more beautiful map plot that you have 7 clusters. I'm gonna move on here. So now we have the data processing standard workflow. I think for the sake of time, I'm gonna skip the doublet finder. It's a quite straightforward process to do it. So right now I'm just gonna introduce what doublet finder is. I'm gonna upload every script I've finished onto GitHub after lunch. So here, what are doublets? So this is a generic process of doing single cell RNA-seq. You have the each RNA trees with the gel bead, and then you have this oiled capsules that we have a gel bead with barcode and UMI and 1 cell that you don't know. But sometimes there will be 2 cells that's been attached to the same unique barcode. So the downstream analysis will call these 2 cells as a single cell. That will be a doublet. And yeah, so there are different types of doublet. There's the homotypic doublets that are doublets derived from transcriptionally similar cells. There's also heterotypic doublets, which means doublets derived from transcriptionally distinct cells. Usually, any tool existed are very bad at finding homotypic doublets, so you would sort of exclude it from the doublet finder. For a doublet finder, there are 3 key parameters. The PN means the number of artificial doublet is usually set to 0.25. And PK is the neighborhood size used to compute the number of artificial nearest neighbor. And the EXP is the number of expected real doublets. So for example, in a data set with 15,000 real cell, a PN of 0.25 would represent an integration of 5,000 artificial doublets. So this means that in total, the RNA-seq captured 2,000 so-called cells, and 0.25 of them are actually 5 artificial doublets. So it comes with 2,000 real cell plus doublet. And a PK of 0.01 would represent a PK of 200 cells. So start with 20,000, and 0.01 would have a neighbor of 200 cells. And here is how doublet finder work. So first step is to have a simulated artificial doublet from existing single cell RNA-seq data, because originally artificial doublet, we are agnostic about that. And we do that by averaging the gene expression profiles of random pairs of cells. And then we merge and pre-process real and artificial data from the simulation. And then here at the third step, we would produce a low dimensional space that describes the similarity between the real and artificial cells. And then we detect the K nearest neighbor using the PK parameter we've just put in for every real cell in PC space. And it is used to compute each cell's proportion of artificial nearest neighbors. And finally, we can then predict the real doublets as cells with the top n PANN value. So this is the proportion of artificial nearest neighbor, where n is set of the total number of expected doublets. So depending on the nature of your data sets, Doublet Finder has provided a table of expected number of doublets you should use for Doublet Finder. So usually the number of cell loaded can be anywhere from 10,000 to 20,000 provided by 10x genome publicly available data. And depending on number of cells covered, you can then choose the appropriate parameter for your EXP value. So for the best practices, it is not suggested to apply doublet finder on aggregated single-cell RNA-seq data. And so, yeah, it's not preferable to run on merged data because different samples may have different proportions of cell type, and merged objects can be large in size. So the previous step, as you see here, would be very, very slow if you have a merged data set. Yeah, so an input data should be cleared of low quality cell as well. So you need to do doublet finder step after all of your QC step. And remove clusters with low RNA UMIs, high mitochondrial reads, and uninformative glycogenes, essentially. So, okay, I'm gonna move on to a more generic step. That's data integration using Harmony and Seurat. Yeah, first, so before in the standard workflow, we've been using one single sample or one data set, but you might want to integrate depending on the scenarios. So there are 4 scenarios of when you should integrate. For example, you have multiple single-cell RNA-seq data sets. You have a treated group, untreated group, knockout, wild type, and different samples. And you might want to do cell label transfer, that is transfer cell type classifications from a reference to a query data set. And there's also integration of multimodal single cell data. I will not go into much detail about this. So essentially, you can integrate single cell RNA-seq data and single cell ATAC-seq data into a single cell multi-omics data set. And so you would integrate signals collected from separate assays. Yeah, so on that, you also have integration of single cell RNA-seq and spatial expression data. You can integrate topological arrangements of cells and tissue with gene expression data. So there are different batch correction methods. I'm going to introduce the canonical correlation analysis within the Seurat, and there's also Harmony. So for the use of Seurat, the goal of the study is to identify distinct tumor cell populations and key genetic mechanisms in HB through single cell RNA-seq. And so in this integration, we have samples from human tumor, background liver, and PDX. So it's a patient-derived xenografts that you implant a tumor sample into a mouse and then you cultivate it afterwards. And they were collected to demonstrate gene expression patterns within tumor and to identify intratumor cell subtype heterogeneity and to define different roles in pathogenesis based on intracellular signal in pediatric HB. And for the goal of our analysis, we're going to integrate data from different patients and correct for batch effect. So I skipped the number 3, that is single cell doublet. I think it's already mostly in there and won't go into much detail of explaining what each step are, but essentially, if you follow the parameters and the steps, it's essentially all up here, and it's I kind of provide some comments. I'll add more comments afterwards. So right now, let's open the integrate Seurat file. Here, essentially, let's move to the data and look at what we are gonna use. So we're gonna use wrong feature here. So we're gonna this is from a publicly available data, and you can find it on the GS with the GSE code. This just gonna go ahead, retrieve it so you can do. Yeah, so it will give you, you can see different samples, 30, 50, and 17. And there's from background, tumor, and patient-derived xenograft. And here I'm gonna do something of for loop in the command line for I. Or we can first create a directory called integrate. And then move everything into and then we can go into integrate to perform our further decompression for I in dot gz do tar CVF XVF $I done. So this is a for loop in a command hack that you can quickly decompress all of them. Okay, we can go back and get the data location. So depending on your naming, I have a list of directories. Integration or integrate. So set to false only. Essentially, I want to write a for loop and merge, input all of the data sets in one run. And then, so quickly check the directories. Here we have all of them here. And so next, let's run for loop for X and directories. First, I wanted the name for each of my sample. Let's do G stuff. And I only want to save this name in my Seurat object. So I'm gonna substitute the filtered feature barcode matrix. Into nothing and assign for X. And then we can read in the counts. So we're gonna use read matrix. And then similarly, when you read in MTX file, feature and cells. For the matrix, we have the data sets. And so depending on where you save the data, and the file name is X. So for X in the directories, then just gonna, it's called matrix TX GZ. And then, paste, we're gonna concatenate them together. Here is the features.tsv and the barcodes. Yeah, sorry. And assign this name we're gonna use in the samples. Create Seurat objects counts. Oh,
Just give it a sense.
you,
See that.
it's a, um. Data sets integration. We have the X. And X. It should- Hmm.
I'll try one by hand, maybe. See if it does. I don't know.
Matrix. Argument two, match multiple.
This feature. Secondary this feature.
Oh, sorry, features.
Features, yeah.
Okay, yes, consult. And then you can sort of like merge all of the data set. Let's call it merge Seurat. Still. So this is what our final merge data sets will be called merge Seurat. And so for this data sets, it's already filtered and processed. So you can see that here are all filtered feature barcode matrix. So I'm not gonna do the QCM filtering step. It's already done if you proceed to view the metadata. And just gonna Seurat cup sample, and we can get it from Pronix. Seurat. Then it sort of split the sample column. Essentially, we'll want to separate by patients, by type, and by treatment group. Okay, so it's finished running. Let's merge. Oh, let's quickly view the Seurat. So here, it's called these different Seurat objects. We can then merge it. Let's use the HB17 background as the X, and then you have the Y to the rest of it. So HB17, go ahead to put all of that in. 17 tumor, and then we have the HB30 PDX, HB30 tumor, and the HB53 background and H. Okay, and within the merge, we can then add. Gonna quickly see what my parameters we have so far. So we wanna assign the cell IDs into each treatment group or each sample. this. So it's start with the 4, 5, 4 to 10. Project. And let's quickly view the metadata of this one. Okay, so here we have each of the name that I added before of the sample name as well as the treatment group and each cell. And here is the metadata. Okay, and let's split them together. So essentially, I want to have an extra 3 columns that will indicate the patient, the type, and the barcode. So I'm going to separate them into 3 columns for the ease of data wrangling in the downstreams. So here we have the row names already. Let's do the separation. Merged. Seurat. Data. Column is sample. We're gonna have another parameter called into patient. The second one is type. The third one is barcode. And for the separation, I use a set. Underline. So we want to separate them based on underline. And then for the merge data, it's essential that we perform the standard workflow. So it's provided here for you. You can just directly go ahead and run them. And with the filtered merge Seurat object, we can check if there's any batch effect by plotting the dim plot to merged. Filtered. And sometimes when you plot it, there's a parameter called reduction. You might want to perform different types of reduction throughout your analysis. So it's important to indicate whether it's UMAP or PCA type of reduction that you've done, so that the function would not confuse patients, because this will have you plot to see if there's any batch effect among patients. And the second plot will allow you to see if there's any batch effect among, let's do type. So type is whether it's being background, xenograft, or tumor cell. And for separation, we can also have different color on the second graph figure. Okay. And this is what I learned like 2 days ago. You can literally just do a straight line here that's gonna have you have the plot side by side. Yeah, this is I didn't know that in R, so okay, and still running. And or you can also do like before that, I usually use this grid arrange P1, P2 call. So it's still running for me, but you can go ahead to put in the code and explore if you see any batch effect in the dimension plot. And yeah, so assume there's batch effect. Let's split the object so that we can find the variable features and select the integration features. So here I have the object list to split the split object, yes. Filtered. And then we want it with, so here I'm just gonna do eliminating or mitigating the batch effects on patient. Let's just do patient. So after we split it, we wanna do normalization and find variable feature for each object for I in so for each object in the list. Data. Also want to do find variable features for each of the object in object list. Step in my oh, I meant you meant this. So we're almost done with the standard QC workflow. Just gonna. Okay, great. So now we have the two plots. So if you literally just plot this, it will show you a side-by-side figure, which is, yeah, I found it very useful. And you can clearly see that there's batch effect among different patients. And for the type, you might need some caution here to call it, is it really batch effect or there's something biologically meaningful going on among the different samples. So here I'm just gonna correct the batch effect for the patient and see how that goes. Okay, and yeah, the grid range will give you the same. Here, let's perform integration to correct for the batch effects. And then we can sort of select the features. Or there's a function called select integration features. That's the tool canonical within Seurat, and have you select the integration features. That's the features it's going to select for batch correction or datasets integration. And then we can anchors, find integration anchors. So this, it used something called canonical correlation analysis within the Seurat. I'm not going to go to detail here, but the paper they have on the integration Seurat have a very detailed explanation of how they do the integration. Scene list equals to object dot list, and we have the anchor features that's selected in the previous step. We named it features. Oh, object dot list. That's list.
OBJ.
Uh,
Oh, no,
this
okay,
one
yes,
is
you're right.
object
You're right.
list. Yeah, it's very confusing to.
No, you're right. So,
Yeah, and this one, I crashed it twice already, so I'm not gonna risk it this time. But essentially with the anchors, you feel free to go ahead and run it if you like have enough resources. So, and then with the anchors, you can safely integrate the data, assuming your computer finished on the previous step. This one is like a pairwise comparison, so it's very, very slow. And the anchor set is the anchors I had before. And then, yeah, so basically you run the scale data for the object, and then you can run PCA and you map for on the Seurat integrated objects. And then after these 3 steps, so I'm not gonna run the rest of the method here. I'm just gonna quickly explain how to assess whether the batch effect is corrected. And then you basically like, you can name it P3 and P4, and then sort of plot it side by side, or you can even do P1, P2, P3, P4 to have a clearer view of whether the batch effect has been corrected. The one time I've succeeded running without RStudio crashing, I've seen that, so in this figure, if there's, imagine 2 by 2, the P3 patient figure after batch effect are sort of well integrated. And here you would use Seurat integrated.
So in this case, P2, oh no, you integrated also the P4, so I was thinking P2 and P4 were the same, but no.
So P2 and
Maybe not.
P4 still looks quite similar,
So similar,
so
not
you would
exactly.
see that even after the batch of correction on the patient sample, there's still distinct cluster within the type. So that'll be for the further downstream analysis. So this is the Seurat integration. Let's move on to the Harmony integration. So before, are we at Harmony? Okay, so before, before moving on to Harmony, I'm just gonna do a quick study design introduction. This is a little bit more simple. So essentially, we just want to integrate on different treatment groups on different types of data. So here it's, you can go ahead and run all of the INFB sample. So if you remember in the beginning of our workshop, we have introduced a RDS format. This is my file after running the Harmony integration. And this is coming from a Seurat incorporated data set and in the library of Seurat data. And you can sort of see what publicly available data is out there. It's all generated in a real clinical experimental setting. And then you can go ahead and install it, load it, see what's like. So, sorry, any questions so far? Here it says no error. Not a valid object. So my IFNB dataset has been successfully incorporated. Let's see what the error message is. I think it's this. So if you run into some warning like this because of the recent version control of the newest version of Seurat, you might want me to update Seurat object because the Seurat datasets has not been updated yet. Let's try if this works. And then okay, so this works after you update the Seurat object. Sometimes you need to step. Just gonna go ahead and rest. So this is very straightforward. I'm just gonna put it in here. We are gonna use tidyverse here to make everything easier. Oh, this is on Mac. If you type Command-Shift-M, it will just pop out this piping. Group. Remember, there's usually the treatment group on any of the Seurat object is called STIM, stands for stimulation. So this is the harmony. It's a little bit different from Seurat, but still does the same job. If you're interested in how it's been done, you can check out the literature. Okay, let's quickly view what it's like in the metadata. So here you have stem, so I put in as a group by variable, and it's already annotated here for the ease of annotation. And yeah, and there's also a meta parameter called reductions. So similarly to what I've said before, there's throughout the analysis, you might want to do multiple types of reduction. So there's PCA we've done, and there's also harmony step on. Both of them are dimensionality reduction. Okay, and then use that with the harmony. There is one function called embedding. It's gonna give you the embeddings from the harmony. You can assign it into IFNB dot embed. Assign it here. And you can sort of check what it's like. This is how I check things, but feel free to use different method if you have. Yeah, you can see how different harmony or it's comparable to PCs, and this is a different cells and how it's been incorporated here. You can do the harmony, but you should still use the harmony object. This is not, you should still use the Seurat object. This is not. options. So remember to specify into harmony, otherwise it'll by default use PCA. Mention equals to 120, then inverse. Yeah, you can all do it in tidyverse. This is just a different way to do it. Yeah, find stars. Let's just specify one that is 0.5 here. To visualize, let's do like a before and after, like before integration and after. So if you go to the before run harmony in the standard workflow step, let's use the plot method to plot this one for filtering. So here I'm gonna specify the reduction into UMAP and group by stem and assign it to uses before the integration and see what before looks like. Filtered. It's not run. Run this step, you usually will get a UMAP. And then like this will be after, right? And this will be using a different Seurat object called IFNB. That's the harmony one. Embedded is not a Seurat object. And function is also UMAP if I run it. And then similarly the group by stem. Okay, let's go back. First cost, okay, great. So I missed it somehow. So yeah, you can see before batch correction, the control group and the treatment group are sort of separated into different clusters. And after, let's plot again using this hack to plot it side by side. You can see that after the harmony integration, the two groups have been integrated well, and the batch effect between the two groups has been significantly mitigated. Okay, so this is the harmony pipeline. I'll upload everything to GitHub after this workshop. And so next step of after the integration, you would find the markers and doing cluster identification. So here I'm going to introduce three main functions. The first one is called find markers in Seurat. You need to sort of specify which two cluster you're comparing between. So essentially, so it's find markers between specific clusters. And then there's also this find all markers. This one is very slow. And if you have a specific biological question in mind, you don't need to run this function, but it's a good way to explore it because it's find marker in a cluster compared to all other clusters. And the final one is more related to our project or research project in general, because usually you have different treatment groups. And so here you have a controlled and treated, and you want to compare them separately between the clusters. You want to know, first question I want to know is whether the gene expression level between the control and treated are different from each other regardless of cluster. The second question you can answer from this is within the treatment group, whether there's difference from different clusters or between clusters within that treatment. So let's start with this cluster identification before moving forward to the automatic cell annotation. Let's go ahead to cell annotation. Yeah, so I think I'm gonna remove everything because I don't think I have enough memory here. Okay, and I'm gonna rerun this metadata. This is the processed IFNB datasets from Harmony. I exported into the dataset link you have. So here we have the stimulations and there's different resolutions and Seurat clusters. Okay, so we can then visualize some of the data. So are people following me still? Show of thumbs, or if you don't have, like, if you didn't follow the previous one, it's okay, because I have the data sets already provided for you. So could you show a thumb up if you're in this step, number 5, cell annotation.r? Okay, awesome. Thank you so much, Diana. Okay, let's visualize the data. Let's call it clusters. So it's already processed and everything, so you don't need to worry about the standard workflow. Reduction equals view map, and then can group by, what's it called? Oh, it's not the metadata. Let's open this as the metadata. It's called Seurat annotation. Seurat. Oh, I want to group it by the Seurat cluster first, not the annotation. Sorry. Clusters, and I want it label to be true. And next is I want to plot the condition by the clusters. Second dimension plot. Here, I want to group by stem. I don't need the labels. There's only 2 groups. And then let's again run the condition by cluster plot. Clusters. So here it's already been integrated, and there are 13 Seurat clusters here. Because it's already been annotated, we can later provide the Seurat annotations. But here I just want to introduce quickly of how to use find all marker. I'm not going to run it. So I'm just going to introduce the different parameters that's used here. And then you can have the log vote change. So this is usually set at 0.25, which is also the default of Seurat. So it's finding all of the markers that have a log vote change bigger than 0.25 compared to all of the rest of the same genes in all of the clusters. And minimum percent set to 0.1, so it's at least expressed in 10% of the cell, this marker. And we can set only pos equals to true. You can, so it's gonna only find the markers that has been upregulated in your specified cluster. You can also set it to false, so it will look for both upregulated and downregulated. We can use DC2 here. I usually use this one, but feel free to use different ones. And we're gonna use the counts. So this is how you do the find all markers. And we are more interested in the conserved markers. So recall that we are interested in to see how genes have been differently expressed among different treatment groups as well as clusters. Sometimes you have a default assay because you not only have the RNA data in, you might have the ATAC-seq, you might have other types, but you can assign it to let the default assay be RNA. Right? And so we can then run the markers cluster. Let's use one of the cluster let's use number 3 here. So if I remember, this one would indicate the B cell, but we can later on plot it. So cluster 3, find the conserved markers. And this is the first identity. So 3, because conserved marker also compared to rest of all of the cluster if you don't specify ident 2 or 3. And grouping variable is the and you can sort of check the head of the markers while it's running. Let's visualize the top features. So we have the feature plot, right? Then harmony features. I'm just gonna wait it to finish running. But the top one is called scRNA-seq. So to plot the feature plot, we want set a minimum cutoff here. So it's based on the quantile. Minimum cutoff is set to 10. So only the first 10 quantile will be light up in the feature plot. If this finish running, I can show you. 1 minute. Yeah, so just to quickly explain what quantile mean. So say you have the seq 1 of 5, and then you can set quantile the Q50. So Q50 of a sequence of 1 to 5 will give you 30, if that makes sense. So it's the 50th quantile, like half. There's Q10 will give you 10. Oh, Q10 will give you 1. So this is quantile means. Yeah, you don't, sometimes, usually the data don't come with annotated cell type. So we can also just rename it because this one is a biomarker in B cells. When you rename it manually, you would sort of do something called rename idents, right? So identity of the cell. So number 3 would be B cell. Oh, sorry. So number 3, Seurat cluster number 3 is CD16 mono. And then you can assign it back to IF. So this will rename the identity, and when you show up here in the Seurat cluster, number 3 will have the name CD16 monocytes. Yeah, again, the cells already have annotations provided in the metadata. So you can set idents as Seurat annotation provided to similarly to set idents. You can just reassign the idents that's IFN harmony into the IFN. The Seurat annotation. Yeah. So this will rename every numbered cluster into their corresponding Seurat annotations. Can again run the plots here with the label to true. Oh, nice. So it's finally finished running. We can look at the head of cluster 3. Yeah, so here is the results of find conserved markers. It's important to know which column stands for. So control p-value is all of the p-value within the control group, similar to the stimulated one. And this one is the average log fold change. It means that this gene has an average log fold change of 4 compared to all of the genes in the rest of the cluster within the control group. And yeah, and this is the adjusted p-value. And so this is the top gene we're going to use. Let's sort of see the feature plot. Again, you can see how this gene have this much fold change different compared to the rest of the cells in other clusters. If we rename it, you can see that it's CD16 mono. Because the cells already have the annotations. So again, this is sanity check, and you can see that it's already been annotated. Okay, so next, let's do the find markers between conditions. I've been harmony. Let's view the metadata again. Oh, there's one cell definition. And created using paste 0. So essentially, we want to combine both the cell type annotation with the treatment group. So it's going to be control B cell, stimulated B cell. The Seurat annotations separated by an underline, and I've been separate. Right? So it's gonna create a new one called cell type condition in I mistyped it. And then we assign it to the new idents. Identity permanently into let's view the metadata again. So here we create a cell type, sorry for the typo, cell type condition into the cell type plus the treatment group. Plot the dim plot again, which will show you an overlapped figure between both the control and the stimulated group. Now, so we're interested in something called so this stimulated group is after the beta interferon response. And then we are going to use the find markers. And then the harmony. The first ident will be again CD16 mono. First one is stim group, and the second identity we're gonna use the control group. Let's see if some of the genes are expressed differently between these two. Do a sanity check if they had to see all of the top markers. And we can also choose one of the markers from the find conserved marker function. To plot it, so we're gonna choose 3 genes. Let's choose this gene that has been found that's differently expressed between this 2 groups. And the top 2 genes of this. You can just play around with to see how different genes have been lined up. This one, and what's the final one? We can do this one. Yeah, but feel free to explore and split by treatment group, and let's set the minimum cutoff again to Q2. Okay, so this is the gene that's been most differently expressed when we run the find marker between the 2 treatment groups on one cell type. Instead of light up in one cell type, we can see all of the cells, regardless of cell type, has been upregulated after the beta interferon treatment. So this is a very typical gene that will upregulate it regardless of cell types after the beta interferon treatment. And the rest of it, you can sort of see there's a relatively consistent expression level before and after treatment group. So you can play around with it. And this is the top gene result from the find markers. You can also try out different genes here. It will, yeah, you can see how it plays after the beta interferon responses. So this is something you can apply in the real research setting for the find markers. Since we have only 10 minutes left, I'm just gonna go quickly of the automatic annotation workflow. So here we were at this step, we found the marker gene. And then depending on the different cell type you were looking for, you can do a manual annotation using a known gene function and annotate the cell by yourself. You can also use a reference single-cell RNA-seq data that's provided in the public, and it'll give you automatic annotated cell cluster. And then you might want to do some manual one after the automatic annotation as well. So there are two types of automatic cell annotation method. One is the marker-based annotation. So it's label cells or cell cluster based on the characteristic expression of known marker genes. There are some typical database that with known relationship between marker genes and cell types. I used a lot of PanGLAO-DB before. And there's also the reference-based annotation that transfer labels from a reference cell or cluster to a sufficiently similar one in query. And we usually can use the gene expression omnibus and cell atlas project and so on and so forth. Yeah, there are some marker-based annotation tool. I won't go into the detail. The strength of these method will assign labels only to cells associated with the known markers, with other cells will remain unlabeled. And however, the markers are not easily accessible for all cell types, and the marker gene or gene set should be specifically and consistently expressed in each cell or cluster, which is a lot to ask. And it worked well once relevant and sufficiently large set of marker genes are collected, and work better for annotating major cell types and may not be effectively distinguishing subtypes of the cells. And there's also reference-based annotation tools where I gonna provide single R for this workshop, and the strength is accurate. It's very accurate of the assigned label, and it avoids incorrect labeling of novel cell types. But the approach is feasible only if high quality and relevant annotated reference single cell data are available. And some tools have low accuracy. So for the single R, I'm not gonna go to detail of how it's gonna be used. So basically we have the step one is identifying variable genes among cell types in the reference set. And then it uses correlation of each single cell transcriptome with each sample in the reference set. And so here it's going to provide a Spearman correlation value of each cell compared to the rest of the cell types. So essentially one sample will have values across all possible cell types, and the highest Pearson correlation, the highest Spearman value would be assigned to that cell. And sometimes if one cell have two cell type assignment with similar Spearman correlation value, it will trigger a step called iterative fine tuning, which reduce the reference set to only the top cell types. If it's not, it's a little bit ambiguous. And just gonna quickly go through the single R step since we don't have enough time. So here we have the reference data. You can go ahead and run all of them here. Oh, it's already 53. I'm just gonna put in what we have for the sake of time being. So here we can use a reference from CellDex. It's a reference index for cell types. And we can use the human primary cell LS data. Expression values are log counts in this reference data set. There's a function called get assay data. Okay, so it's in with all of the QC step and standard workflow done. And this is the dim plot, and this is the metadata of the filtered Seurat object, PBMC. So next, let's get the assay data. See, wait, let's use the filter data, then the slot to counts. So we're always recall there are 3 different parameters: counts, datas, and captures. So we're using the raw counts here. Prediction. So this is a 1-line single R and automatic cell annotation. Counts. The reference equals to the reference we inputted, and the label is equals to. There's a label dot name. If you don't have single R, you can use the bioconductor manager to install it, as well as Soundex. I love this, yeah. And let's run single R again. And you can check the prediction by using just calling it and PBMC. Filtered. It will create a parameter called single R. And you can assign the labels to the prediction labels. That's match the row names. PBMC Seurat filtered metadata. In the row names, the prediction. So it will essentially assign all of the predictions done by single R to your Seurat object. You can check the dim plot with the newly annotated Seurat object. So this will show you the annotated clusters for your data sets. However, it is very important to assess whether the automatic annotation tool have been performed correctly. So we have different methods to assess whether your annotation tool have reliable results. So you can first look at the scores of it. You can also plot a score heat map. That's hopefully it can finish run for the first set of assessment. I'll upload everything onto GitHub. So it's still already there. You can see all of the scores here, corresponding to each existing cell type here. It's a very long score. We have the scores. Match. Running.
Is it meta.data?
Yes. Yeah, feel free to go have lunch. I'll try to have this explained at the end of the workshop, but it's there. Okay.
Oh, is it
It's
a round
a-
parenthesis? Yeah, right.
Back.
Yeah, that's. the problem.
Okay.
That's the problem.
Yeah, it's always the stupid bugs that keeps you stopped at the middle of your coding. Okay, great. So we have the single R labels. It's provide like somewhere between 12 cell types. And to assess the reliability of this, you can see that the prediction have here. Let's plot the scores. So here, the first cell have 10 scores. Here, so it's not like a lot of 0.9. You want to look for cell assignments that are not ambiguous. Similarly, we can also just plot a score heat map to see. Okay, this is much better. So here are all of the labels that has been assigned by the automatic annotation tool, and each column, like each little line here, represents a cell. Okay, so in order for the assign to be non-ambiguous, you want to look for the scores that has been very high for one cell type assignment and relatively low for the rest. So here you can see some of the ambiguous cell assignment. And this is just one way to assess it. You can also based on deltas. So deltas is how the first score are different from the rest of this score. Just gonna put all of them here. You can check it on YouTube later. To go, this is just plot delta distribution. And this shows how the scores are different from the rest of the scores. So you want to have a very high value. And if there are ambiguous assignment, it will trigger the single R to do fine pruning if you allow it to. So this is true. And you can see that how delta have been relatively low in these cell type assignment stuff. And finally, it's just another 2 lines of code. This is can create a table of assigned equal to prediction labels. And the clusters equals to PMC Seurat filtered. This is the Seurat clusters we have is a number. And then let's see the tab really quick. Okay, so it's associated with different numbers of different number of cells that have been assigned into this cluster. And there's a P heat map. Let's do a log transformation because a lot of the clusters have 0 cells assigned into this. 10, just tap +1 and color. Pellet. So the lowest one would be white. The highest one would be blue. And we want 10 values in between. And I forgot to load the libraries. Okay, so each column is a cluster. You can see how some of the clusters have been very distinctly have the monocytes. All of them, most of them are monocytes in these clusters, but some of the cluster have ambiguous cell type annotations between monocytes, T cell, B cell, as well as predecessor B cell, CD94 lemons. So this is it'll kind of call for some of the downstream analysis or manual annotation that you can go check online, publicly available data sets, and put in the markers you found using the find markers, find conserved marker functions in it. So yeah, so this is the end of the workshop, and this is just a pseudo book differential equation. And throughout, we've covered number 1 to 6. That is reading in different types of data, the data processing standard workflow, and I'll upload the Doublet Finder function pipeline in it. And we try dataset integration using different Harmony and Seurat functions, find markers and cluster identification, and automatic cell annotation. I hope this is somewhat useful for you, no matter in your side project or your main research project on using Seurat for all of downstream analysis. Thank you for joining me very much.